All right, well, good afternoon, everyone. It's my distinct pleasure, my bittersweet pleasure, to have the opportunity this afternoon to introduce Justin Conover, who's here for his doctoral defense. A little bit of history. Justin first came up to visit Iowa State and spent a summer in my lab. I don't know what year that was, around 2013. For a research experience for undergraduate program. And he got really turned on by the science. We were really turned on by his presence. And then he applied to graduate school here. And we were very quick to be delighted to accept him. I went back to his graduate application file and to see what he wrote and to see what his referees wrote, his letters of reference. And with his parents who are here visiting in the room, you might appreciate his comment. I don't know if you saw his application, that when he was a little child, he was indeed a nerd. And in third grade, he was doing logic puzzles and probably thinking about science. And I went back to my letter of recommendation for Justin, and I wrote that he has extraordinary scientific potential. Well, it turns out I was prophetic. Justin came and he has spent a long journey here and has explored and expanded our science, during which time he has published, and I hope everybody out there in Zoom land is sitting down. He has published 18 papers. Just 2 this week were accepted with minor revision, both in the very top journal in the field, Molecular Biology and Coevolution. I'm pleased to report. So everything in his thesis will already, let this be a model to other doctoral students. Everything in his dissertation will already be published. So he's been extraordinarily productive scientifically, but it wasn't just that, he's also been active in grant writing. It's too bad he can't put co-PI on his CV for the $2 million National Science Foundation Plant Genome Program Award we have, which he participated in developing intellectually and in the writing, but as a graduate student, he had to be sort of a hidden person behind the scenes. But he's truly been an integral part of this work and will continue to be so after the completion of his thesis. So he's been accomplished in fundraising in many activities outside of the academic sphere, including running. I managed to hoodwink him into running the Beer Lovers Marathon in Belgium with me a few years ago. He did finish. He was smiling at the end, either from the beer or having finished the marathon.
It was both.
One of the things that we do as major professors is train our students scientifically, but also in many other aspects of being a professional and learning the sociology of science and codes, best practices, codes of conduct. And Justin, a bit more advice to you at this time. I think it's not really considered appropriate on your curriculum vitae to say, "We'll run for beer." So I'm really going to miss Justin when he leaves. It is true that I will cry in my beer when Justin does leave. And so with that, I'd like to introduce the topic of his dissertation and the title, which is Molecular Evolution Following Allopolyploidization in Gossypium. And Justin, the floor is yours.
Thank you so much, Jonathan. Let me just correct this camera real quick so that you all can see me. Oh, sure. Thank you, Jonathan, for that very generous and slightly embarrassing introduction. I really appreciate it. As Jonathan mentioned, I will be talking today about molecular evolution following allopolyploidization in Gossypium. And Gossypium, of course, is the generic epithet for what we now call cotton. Before I get started, though, I want to acknowledge that this research was conducted at Iowa State University, which is a land-grant university that was built upon the ancestral homelands of the Iowa people. And secondly, I want to acknowledge some of the societal impacts that my study system has had. If my slide advancer will work. There we go. So in Sven Beckert's book, which is a really great book if you guys are interested in cotton and the production of cotton and the history of it, he says that cotton is as familiar as it is unknown. Even the graduate students on this Zoom call who may be still in their pajamas laying in bed are still surrounded by cotton. And cotton fulfills, I mean, it fills our lives. We're constantly surrounded by it. And yet we know so little about how it's actually made, how it's grown, and the impact that it's had on society. Cotton was the first main crop that led to capitalism being expanded into a global enterprise. And the products of Indian weavers paid for slaves in Africa that worked on the plantations in the Americas, whose agricultural commodities were sold to European consumers. And so cotton is really the first major crop that was that had this trade that spanned 4 different continents. And this has led to what we know as global capitalism today. In Sven Beckert's book, he refers to this first rise of global capitalism as war capitalism, and notes that it was in part characterized by a really dark past, including imperial domination, the expropriation of vast territories, decimation of indigenous peoples, theft of their resources, enslavement, and the domination of vast tracts of land. And of course, it's comforting to think that this is all in the history of cotton production, but as we know, history has consequences, and unfortunately, racism didn't end with slavery. And as much as our individual incentives or our collective desires for objectivity may pursue, science cannot happen outside of a societal vacuum. In this paper from 2020 that looked at a survey of PhD and master's students from 2017, people of color, including both Black and Hispanic people, are severely underrepresented in the biological sciences, especially in the fields most closely related to what I work in, which is ecology and genetics. And according to Ibram X. Kendi's book, How to Be an Anti-Racist, the only way to undo this racism is to consistently identify and describe it and to dismantle it. This book by Edward Baptiste is an attempt at retelling the story of slavery based on the slave's perspective, and is a really enlightening and really difficult book to read. But I also want to open it up to everybody who's listening to think about the parts of our science that maybe we're not aware of, what parts of the history of our study system we may not be aware of. For instance, I work in genetics, and in some of the earlier courseworks I had to take as a graduate student, we read about HeLa cells, which were cancerous cells stolen from a Black woman named Henrietta Lacks. And it wasn't until several years after her passing that it was known where these cells came from. And her nor her family have still received any sort of compensation for her vital contributions to science. And secondly, there's a lot of crops that are endemic to the Western Hemisphere that are also deeply entrenched in racism and colonialism. And although cotton may be the most intricately linked to these horrible practices, a lot of crops that are native to the Western Hemisphere have this problem as well. And so I just want everybody to think just for a second about how can we elevate and center the lives of those, or center the voices and lived experiences of those most impacted by our study system. Okay. So I want to share a little bit about my journey into science, which starts in a small town called Pattonsburg, Missouri. For those who are in Ames, if you go out to I-35 and take a right, you'll eventually hit Pattonsburg. This is I-35 in the lower right-hand corner here. It's a town with a population of less than 350 people, and I had 13 people in my graduating class. And so this town is really focused on agriculture. The big white bubbles in the background are actually the school, and right across the street from the school is a field that rotates between corn and soybeans. And in fact, my childhood was so ingrained in agriculture that I thought I should share a really embarrassing photo of myself from 2008, where I actually placed at the Missouri Sheep Producers Association public speaking contest. So the question probably arises, how did I get from agriculture into evolutionary genomics? Well, it started at Missouri State University in Springfield, Missouri. And as Jonathan mentioned earlier, in 2013, I was looking for internship programs and found this program through the National Science Foundation that I could apply to all over the country. And as my post from Facebook says, I filled out applications in Washington, and Delaware, and Michigan, and I maybe could have ended up in Iowa, except I was kidding about that Iowa part, but joke's on me because Iowa was the only place that accepted me. And this was a fantastic summer experience. It fundamentally changed the way that I view science and has fundamentally altered my career trajectory. There were 10 students from all around the country, including one from American Samoa, who joined that summer. And I worked in Jonathan Wendel's lab that summer studying the evolutionary history of cotton. This picture is of me and Rick Masonbrink, who at the time was a postdoc in the lab. And this picture was actually taken in this room during a defense of a former postdoc of Jonathan's now. Yeah. So that summer that I spent studying cotton, I kind of expected to talk a lot about cotton production because that's the only aspect of cotton that I knew. But they kept talking about cotton as a model to study this thing called polyploidy, which I knew nothing about. And so I first want to explain what polyploidy is. So here, if we look at a karyotype of a normal diploid human genome, in this case, a male, we see that there's 2 copies of every chromosome, 23 pairs of chromosomes. But if we look in a cancerous leukemia cell, we can see that there's 4 copies of every chromosome, and this is considered a tetraploid. And although polyploidy events like this are normally detrimental in most animal systems, although there's increasing recognition that some animals may be able to tolerate this more, this mostly leads to cancerous and really bad things in animal systems. But in plants, we find it all over the place. We find it in tobacco and in peanuts and cotton, and perhaps my favorite tetraploid in coffee. And these all have 4 copies of every chromosome in their genome. But it doesn't have to stop 4 copies. You can also have 6 copies, as is the case in wheat, or you can have 8 copies, as in the case in strawberries. And you may notice that all of these are an even number of complete chromosome sets, and that's because if you have an odd number, then you get failure of meiosis, which leads in plants to the failure of seed development. And it's this case of triploidy that's allowed us to have seedless watermelons and bananas that don't have any seeds in them. So although it's been known for a while that polyploidy is common in plants and in species that we currently recognized, it really wasn't until we started sequencing genomes and comparing one genome to the other that we realized how prevalent this polyploidy actually was. If we took, for example, the human genome and compared it to its most recent common ancestor, or to its most recent relative, the chimpanzees, you can see a really strong linear relationship between one chromosome and the other. You can see that in chromosome 2, there was a fusion event where the chimpanzees had 2 chromosomes, but now they've been fused into 1 in humans. And you can also see areas that have been flipped upside down, which are those triangular areas on the screen. But whenever we started sequencing plants, things got a lot more complicated. For instance, one of the first 2 plants to be sequenced were maize and rice. And the genome changes that occurred between them were so complicated that we even had to use a circle to depict what these relationships look like. So on the bottom right-hand corner here, we can see the different colors for each of the rice chromosomes. On the outside are the 10 maize chromosomes. On the inside is the relationship to those 10 chromosomes to the rice chromosomes. And they're colored by the depiction in the bottom right. And these weird arcs in the middle refer to duplicated regions, where you can find a string of genes on one chromosome that show up in the exact same orientation somewhere else. And so sometime in the evolutionary history of corn, there had to have been this genome duplication and then followed by chromosome downsizing and massive amounts of gene loss. And it turns out maize isn't unique in this aspect. Once we start sequencing more and more plants, we're finding more and more polyploidy events. I think now there's over 100 and nearly 200 ancient events that have been identified. And a lot of these have happened around 65 million years ago, which coincides with the last mass extinction event, where over 70% of planet animals went extinct, including non-avian dinosaurs. And so it could be the case that polyploidy offered some sort of advantage to this really rapidly changing environment. I found this diagram from a few years ago that also has ancient polyploidy events depicted on it. And if you look at cotton here in the underneath the red arrow, you'll see this gray star, which means that there was a polyploidy event, but the details of that were unclear. And I should have taken this as a hint that this was going to be a difficult problem. And this is actually the first chapter that I'll talk to you about. And actually, as a result of that chapter, the details may even be more unclear than what we had initially thought. Ancient polyploidy events are so prevalent, in fact, that only 2 plants have been sequenced that don't have any polyploidy events outside of the event that predated the evolution of the angiosperms. And this second genome was only published on September 2nd of this year. And so there's very few examples of plants where polyploidy is not important in understanding its evolutionary history. So the outline of my dissertation, the first part of my talk will be a bit of a mouthful of a moniker for a manuscript. And that chapter title is A Malvaceae Mystery, a mallow maelstrom of genome multiplications and maybe misleading methods. This is a paper about the macroevolutionary aspects of polyploidy in the Malvaceae, and it was published in the Journal of International Plant Biology in 2019. The second chapter out is a new method that I developed that was published earlier this year called pSONIC, and it stands for Ploidy-aware syntenic orthologous networks identified via collinearity. The fourth chapter is the focus of how polyploidy may act at the population and species level. And this deals with deleterious mutations, and I found that they accumulate faster in allopolyploid than in diploid cottons and unequally between subgenomes. And finally, the last chapter is how polyploidy may impact evolution at the cellular level, where as part of this NSF grant that Jonathan mentioned earlier, we've been able to look at coevolutionary forces that may be acting within a single cell. As Jonathan mentioned, on Monday, I found out that chapter 4 would be accepted for publication in Molecular Biology and Evolution. And on Wednesday, I found out that chapter 5 will also be published. And so that's really exciting to know that after the Graduate College accepts my dissertation, which is not an easy feat, and after my committee accepts it, which is maybe an even harder feat, there will be very little work that I'll have to carry on into my postdoc from this. Before I get talking about the second chapter, though, I want to acknowledge that because polyploidy has such a profound effect on so many areas of biology, that we've kind of had to create a whole new set of jargon in order to describe what's actually happening in a polyploid. And so first, I want to talk a little bit about this foreign language of polyploidy. So first, an ortholog are any 2 genes where if you were to create a gene tree, you would find that they would be separated by a speciation event, whereas a paralog are any 2 genes that are separated by a gene duplication event. You can also use an umbrella term of a homolog to describe both paralogs and orthologs. And we can also use this KS value, or this synonymous rate of evolution, to kind of time when these gene duplications events occurred. Secondly, I want to distinguish autopolyploid from allopolyploid, auto meaning same. And so an autopolyploid is just a polyploid that arises from the same species, perhaps even within the same individual, whereas an allopolyploid formed from 2 different species. And finally, a subgenome in an allopolyploid is just one complete set of chromosomes in an allopolyploid. And so, for example, a allotetraploid would have 2 different subgenomes that we would refer to as. So the Malvaceae family is a quite diverse family that has a lot of really important ecological and economic plants in it, including Adansonii, or the baobabs, which are these famous giant trees in Africa, with these giant trunks and little tiny leaves on top. Hibiscus is the plant that you may see planted in your garden, and it's an excellent example of these exerted styles and stigmas and monodelphous stamens. Also, cotton, as you all know, is the topic of my dissertation. Durio is a crop that is native to Southeast Asia, and it's actually banned in public transportation because it smells so bad. And it's been described as turpentine and onions garnished with a gym sock. And so this crop is also really important to Southeast Asian communities. And then finally, there's this plant called Theobroma, which is one of the more distantly related plants to cotton. But you guys may know this plant instead as chocolate. I don't know. It comes from the Malva genus is the type specimen. Probably because the Malva was the first to be described there. I'm not sure where the name originates. That's a good question. So when the first cotton genome was sequenced in 2012, which was Gossypium ramundii, they compared this genome to the chocolate genome and also to the grape genome. And they found that for a lot of segments that occurred once in grape, it occurred once in chocolate, but it occurred 5 or 6 times in cotton. And so this is evidence that there was either a 5 or 6-fold multiplication event that happened somewhere in cotton's evolutionary past. Separately, in 2017, the genome of Durio zebethinus was sequenced, and they found in the upper left-hand corner, you'll see the 10 chromosomes of Theobroma, and they roughly each of those chromosomes roughly matched to 3 of the chromosomes in the Durio genome. And so sometime along the Durio evolutionary history, there's been a whole genome triplication event. And so the research questions that I was interested in is, one, when did these ancient polyploidy events happen? Was it one event, or were they independent events? And secondly, what other subfamilies in the Malvaceae are descendants of these duplication events? So this paper was done in part with Dr. Nisa Karimi, who's now a postdoc at the University of Wisconsin at Madison. And we were both co-first authors on this manuscript. And we sampled 8 of the 9 subfamilies in the Malvaceae, all diploids. And so the cotton that we used here actually isn't the cotton that you would normally think of that has agricultural purposes, but is a close relative. And we used published chloroplast genome sequences because they are all maternally inherited, and they're a lot easier to work with than the nuclear genes we may have. We also used RNA-seq along with the published genomes that were available to find which of these subfamilies had a whole genome duplication event. And so here we see these KS plots. And if you see a peak in these plots, then that means that there's been a whole genome duplication event sometime in its evolutionary past. Here you'll see that the top 2 species do not have a peak, and so they don't have evidence for a polyploidy event. And also the 4th species down, Dombea, does not have evidence for a polyploidy event. And so it's kind of odd that these species that have evidence of polyploidy don't form a monophyletic clade within the family. And so we further investigated whether this was due to perhaps incomplete lineage sorting in the chloroplast phylogeny, or perhaps this was actually a hybridization event via allopolyploidy that formed one or both of these clades of polyploids. So the first one that we did was we tested whether this was just due to incomplete lineage sorting. And the way that this test works is if you have any tree topology with 4 samples, there's 3 different orientations of those 4 samples on that tree. One in which A is more closely related to B, one in which A is more closely related to C, and one in which A is more closely related to D. If we take a sampling of a bunch of nuclear genes and we see that all 3 of these tree topologies are equally likely, then that's evidence that there's probably just incomplete lineage sorting along this phylogeny, and a simple bifurcating tree can explain the accurate origins of this history. If we see that only 2 of the trees are best explained and the other one is obviously worse, then that could be evidence of some sort of hybridization within the clade. And finally, if only one of these trees is best, then that's representative of the species tree. And so we used a program called Bucky to do this Bayesian concordance analysis using over 1,000 single copy genes in five of these species in the family. We had whole genome sequences for Gossypium, Bombax, Durio, and Theobroma, but because Dombea was so important in the phylogeny, we decided to include its transcriptome in this data as well. And the numbers down here, the number below the line is just the coalescent units, so that's similar to the branch length. And then the number on top is the Bayesian concordance factor, which you can think kind of as a proportion of the genes that fit that given tree. And so for the node that links Gossypium from Bombax, it's really close to 1, and so this is the true species tree topology. But for this more internal node linking Durio and Dombea with Gossypium and Bombax, the number is really close to 1/3, which is what we would expect under just complete, incomplete, or under total incomplete lineage sorting. And so we came up with this this hypothesis where Durio may be the result of some sort of hybridization or allopolyploidy with Dombea, and perhaps there's incomplete lineage sorting mixed within there. And then the whole genome duplicate or the whole genome multiplication number of Gossypium, we don't know if it's 5 or 6x from this hypothesis. Secondly, we wanted to ask if there was evidence directly for allopolyploidy in this clade. And the way that this test works is if polyploidy is just a result of autopolyploidization, then you should see just a simple bifurcating tree. But if there's an allopolyploidy event, then you should see some lineages are more closely related to a given species, and they don't form a monophyletic clade like we would expect, as what's shown in the bottom portion of this figure here. And so we used a program called GRANDPA, which stands for Gene Tree Reconciliation Algorithm, with mole trees, which are multiply labeled trees, for polyploid analysis. And what we found is that there could be evidence for an allopolyploidy event that led to the rise of either the ancestor of Gossypium and Bombax in the blue circle, or of the ancestor of Gossypium, Bombax, and Fermiana in the blue and green circle. And this would be the maternal progenitor, because this is the chloroplast phylogeny, which is maternally inherited. And we think, and this has evidence that the other donor for this allopolyploidy event occurred somewhere along the blue or the green branch lengths. And so the second hypothesis that we have is that there was a whole genome triplication event that occurred in the evolutionary history of Durio. There was an independent whole genome duplication event that happened perhaps in the ancestor of Fermiana, Gossypium, and Bombax. And then there was a subsequent hybridization between this hexaploid and tetraploid to form what we now see as a 5-fold multiplicated cotton genome. So overall, we have 2 possible hypotheses, although these are in no way the only hypotheses that are possible. And we still don't know whether this whole genome multiplication event was a 5 or a 6-fold multiplication. And unfortunately, this question is going to be really difficult to answer until we get genomes of higher quality for more representatives within the family, and until we have methods that can deal with these higher level ploidy levels. For chapter 3, I want to go into a little bit more of this foreign language before I describe the new method that I developed. And the first one is an orthogroup, which is just the collection of all the orthologs and paralogs in a set of species that are descended from a single gene in the most recent common ancestor. So in this example here, both A, C, D, and X, Y, and Z would all be considered one orthogroup because they all descended from a common ancestor at the top. Also, synteny is how we describe a conserved gene order. So in this case, in chromosome 1 of humans, and if we compare it to chromosome 1 of chimpanzees, there's a really clear syntenic relationship between these two chromosomes because you can easily go down the sides of the chromosomes and find the same genes in the same order. And finally, a homolog is what we call these duplicated genes in an allopolyploid. Because they're still technically separated by a speciation event, they're not orthologs, but because they're duplicated, they're also paralogs. And so the polyploidy community had to create this new term to describe the relationships between these genes in a current allopolyploid. So I developed a program called pSONIC, which asks a really simple question, which is, can we use synteny to infer orthogroups instead of sequence similarity? Normally, if you want to infer a group of orthogroups for distantly related species, you have to rely on how similar those genes are to each other. But because we're sequencing more and more species that are really closely related to each other, synteny is a really powerful way to infer orthology instead of just sequence similarity. And of course, the problem in doing this in plants is that even if we have the 4 species here that we can find really clear syntenic relationships for, there's also all this other trash from ancient polyploidy events that we don't want to include as genes in these orthogroups. And so the main challenge of pSONIC is to filter out which syntenic groups are the good ones and which ones are the bad, and exclude all of those that are bad. So this is a program that's written in Python, and all the code is available on GitHub. It can be used for any ploidy level, including if you're only working with simple diploids and there's no need for this ploidy aspect. It's still perfectly usable in diploids. And when we used it in cotton, we found that it places between 75 and 90% of genes, which is a pretty good rate. 59. We also found that it created 59% more single copy orthologs than OrthoFinder, which is kind of the leading program in the field that people use to find these orthogroups, and that 99% of the single copy ortholog groups have the correct tree topology that we would expect given the known species relationships in cotton. For my 4th chapter, I wanted to look at deleterious mutations and how polyploidy may impact how we view population genetics. So deleterious mutations through PopGen are well known to be controlled by a few different phenomena. One is the effective population size. So populations that are larger can purge these deleterious mutations more efficiently than smaller populations can. Increased recombination rate can also increase the efficiency that these deleterious mutations are purged, because you can link deleterious mutations together and more efficiently purge them. Life history also plays an important role. So species that are asexual tend to accumulate deleterious mutations faster than species that are sexually reproducing. And finally, dominance can play a role too, or how much of a fitness cost these mutations incur if they're only present in one copy instead of in both copies of a diploid. So the role of polyploidy isn't in deleterious mutations is an old one. JBS Haldane was talking about the effects that epistasis may cause in determining the trajectories of mutations. And he said that among other things, it will favor polyploids, and particularly allopolyploids, which possess several pairs of sets of genes, so that one gene may be altered without disadvantage, provided its functions can be performed by a gene in one of the other sets of chromosomes. And this theory has been used for a long time to explain how genes can gain new functions after polyploidy, because as long as one gene retains its ancestral function, the other gene is free to mutate and gain a new function, or be used in a different part of that organism, or just be lost altogether. But this turns out to be a really difficult prediction to actually prove because there's a lot of complicating factors. So to test this, I decided to use cotton, obviously, to test this hypothesis. And in cotton, the 2 diploids that gave rise to the allopolyploids are Gossypium urbundii, here in black, which originates in northern Peru, and Gossypium herbaceum, which the wild form is native to southern Africa. About 1 to 2 million years ago, a member of the A group, which is Gossypium herbaceum, traveled to the Americas and hybridized with a member of the D group that's closely related to Gossypium remundii, and formed this clade of allopolyploids that have since diversified into 7 different species. Some of these species are native to Northeast Brazil, including, and one of them is Gossypium barbadense, which is also known as Pima cotton or Egyptian cotton. And its close relative is found in the Galapagos Islands, and it's properly named Gossypium darwinii. There are also wild cotton species that are found in Hawaii. And then finally, Gossypium hirsutum is this species that we all think of when we think of modern cotton from an agricultural perspective. And it has 2 very closely related species, one that's located in Hispaniola, Gossypium eckmonianum, and one that was just described in 2017 at an Air Force base in the Wake Atoll, which is named Gossypium stephensii. And so I used whole genome resequencing data from all of these species except for Gossypium barbadense in green here, because we don't have any wild versions of this. And so that avoided any complications that may be due to domestication instead of just simply polyploidy. And use some comparative genomic techniques to determine which SNPs were deleterious and which SNPs were not. So the way that these methods work is if you have a whole genome alignment, in this case, this is using a bunch of mammals to do a whole genome alignment, you can estimate the neutral rate of evolution at these synonymous sites in the genome. And so they shouldn't be affected by purifying selection at all. They should be controlled simply by genetic drift. And if there are any sites in the genome that evolve slower than these sites at the neutral sites, then we can infer that there has been some sort of selection trying to get rid of them from the population. Otherwise, they would be just as frequent as the neutral sites are. And so, for example, here, the column that's outlined in red would be a deleterious site if there were a change in that position, because it is very conserved across this mammalian phylogeny. And these methods can be used for any gene or genome sequence. And so this isn't something specific to cotton. You can use it for any species of interest that you have. Here we go. So first, I want to look at synonymous mutations. My PowerPoint will behave. Here we go. So first, I want to look at synonymous mutations that have arrived or that occurred since the divergence of each diploid from its polyploid progenitor. And it's important to note that these phylogenetic distances are not equal. So although it looks like these 2 groups are very different from each other, we can't make a fair comparison because we would expect that. The most important thing to pay attention to in all of these figures for the next 2 slides is comparing the red and black dots, which indicate the 2 diploids, from all of the other dots, which indicate all of the polyploids. And the geographical locations are still placed in the upper right-hand side for you to remember. And so using almost 9,000 homologous pairs that were identified by pSONIC, which means that they have conserved syntenic relationships, and we only use homologs that had less than 5% difference in annotation length. So for synonymous mutations, we don't see very much difference between the diploid and the polyploids. But when we turn to non-synonymous mutations, we do see quite a bit of difference. I also used 2 different methods that were similar to what I described before to identify which SNPs were deleterious and which were not. One of them is called GERP, which looks at selection acting on a nucleotide level, and we still see a difference between the polyploid and the diploid. And the other one is called bad mutations, which looks specifically at changes in the protein sequence. And again, we see this really strong evidence that there is a difference between the diploids and polyploids. Also, if we look just at those SNPs that are segregating within the polyploids, we can see that there's a lot of variation between the different species here. For example, the purple dots, which represent Gossypium tomentosum, which is found in Hawaii, we found that there's a lot more deleterious mutations here. which could be indicative of demography having an effect on some of these findings that we have. I also wanted to look at what proportion of all of these non-synonymous mutations are actually deleterious to see if these are higher in the polyploids than in the diploids. And it turns out that they are. When we look at the mutations that have occurred since the divergence of the diploids from the polyploids, we see that there's quite a bit difference between each diploid and its respective subgenome, which means that polyploids accumulate deleterious mutations faster than diploids due to this masking effect of having 2 copies of every gene in the genome. And secondly, if we look at just the mutations that have arrived since polyploidy, these now all share a most recent common ancestor that's not confounded by different phylogenetic depths. And so we can actually look at differences between the subgenomes here. And we find that in the AT subgenome, deleterious or a larger fraction of the non-synonymous mutations are deleterious compared to the DT subgenome, which means that this masking effect may be acting unequally between the 2 subgenomes. And also the bias could be species specific. For example, if you look at the yellow dots, there's a lot less difference, only about 1% difference between the AT and DT subgenomes. Whereas if you look at any of the other dots, there's at least 2 or more % difference between them. And also the island endemics have a higher load than the species that are not endemic to islands. So I mentioned earlier that demography could cause these sorts of patterns. And so I wanted to try and find a way to explicitly exclude demography from this explanation and to figure out if this pattern was due to demography or if it was actually polyploidy. And so we can set up some expectations if this is due to demography and compare it to what we actually see in the data. So in population genetics, you can measure the deleteriousness of a mutation ranging from 0, meaning that it's completely neutral and it has no negative fitness consequences in the individual, all the way up to 1, which means that it's lethal. And there's also this value of 1 over the effective population size. And the nearly neutral theory predicts that if S is less than 1 over NE, so if it's on the left side of this red bar, then those mutations should be controlled solely by genetic drift. And if the deleteriousness is to the right of the red bar, then it should be controlled by purifying selection. It should be removed from the population. And for example, if you have a population bottleneck, which means that NE gets smaller, then the number of mutations that are deleterious, more of those will be controlled by genetic drift. And if you have a population expansion, then a larger purifying selection will be more efficient at purging those mutations from the population. And so regardless of what this demographic history may be, we expect that for strongly deleterious mutations, there should be no difference between ploidy levels. The strongly deleterious
Hello. Zoom must have crashed for him.
Somebody text Jonathan or somebody like that. Does anybody have his phone number? So that they know it's
I don't
done.
know.
This is John. I can text him.
They know it's off. I just looked over in the room and they're working on trying to fix it.
Okay,
Oh,
sounds
okay.
My apologies, everyone. My laptop just died, even though I thought I had it plugged in. So we're going to get this switch over to a new laptop, and we'll be back in just a second.
Laptop, or it would have, I guess. Sounds like they're coming. Oh, there you are.
Oh, me?
We're back.
We're back.
Are we back?
I think so. Okay. Are you ready to go, Matt?
We're getting exactly a little over here.
Yeah.
Good to go. Okay. I'm going to back up a little bit because I'm not sure exactly where I was cut off. So I wanted to see if these changes that we saw between the diploids and polyploids could be due to demography. And although there's really low genetic diversity in cotton and this polyploidization event happened a long time ago, we can't directly model it. We can set of expectations that we would expect to see under demographic shifts and compare that to what we actually see in the data. So in population genetics, we can measure the deleteriousness of a given mutation between 0 and 1, where 0 is completely neutral, meaning it has no fitness consequence to the individual, and a deleteriousness of 1 is lethal. And so that's immediately purged from the population. There's also this value of 1 over NE, which is 1 over the effective population size, which is just how big the population is. And the nearly neutral theory of evolution predicts that mutations that have a deleteriousness that are less than 1 over NE, which would be on the left side of this red bar, would be controlled by genetic drift, and mutations that are greater than 1 over NE, or on the right side of this red bar, would be controlled by purifying selection and removed from the population. And so if we have a population bottleneck, then 1 over NE increases and more mutations will be controlled by genetic drift. Whereas if we have a population expansion, then purifying selection controls a larger set of the genes, even those that are more weakly deleterious. But regardless of what this demographic scenario looks like, we expect that strongly deleterious mutations should not change between ploidy levels. Deleterious mutations should be purged if they're strongly enough in both diploid and in polyploid. And those mutations that are mildly deleterious would be the most variable. So depending on whether we see a population bottleneck or an expansion could influence whether there's more or fewer deleterious mutations that have accumulated. What we actually see in our data, though, is a trend that's very opposite of those predictions, where we see that when we compare the relative number of deleterious mutations for mildly, moderately, and strongly deleterious, we see that actually strongly deleterious mutations accumulate faster than moderately deleterious mutations, and moderately mutations accumulate faster than mildly. And sometimes this is over 50% faster. If you look at 83, Gossypium tomentosum, again, that Hawaiian species, it accumulates strongly deleterious mutations almost 50% faster than the diploids do. And so not only was this exciting because it was the first confirmation of this hypothesis from 1932, which is almost older than sliced bread, but it means that I can add a new addition to this foreign language to describe this effect in a population genetic framework. And I've just decided to describe this as this homologous epistatic dominance, where it's not just the dominance of a mutation that controls its fate. It's not just the deleterious of that mutation, but there's some sort of interaction between the homologs that can alter how these mutations accumulate within a population. The final chapter is about cytonuclear coevolution. So if we look in a general diploid, if we have this picture of a traditional cell in the middle, we know that there are a lot of mitochondrial chloroplasts that are in the cytoplasm, and in the middle of the cell is this nucleus, which is where the genome is located. Within each of these mitochondrial chloroplasts, there are these genomes that contain a small number of genes that are used in the mitochondrial chloroplast, but are nonetheless important in the functions of mitochondrial chloroplast, but they can't function on their own. There have to be genes that are encoded in the nucleus that are transported to these mitochondrial chloroplasts, and they work together with the genes that are in these organelles to form all the protein complexes and all the proteins that we see in these organelles. And we also know that these genes can be transcribed at different rates as well, and there can be correlation between the rates in the mitochondria and in the mitochondria and chloroplast and in the genes in the nucleus. If we look at a polyploid cell, though, polyploid cells tend to be bigger, and we think that there should be more chloroplast and mitochondria per cell because the cell has more needs for bioenergy. And there can also be more genomes per organelle. So there can be more genomes per mitochondria and per chloroplast in these cells. And again, there could be interesting correlations between expression levels of these genes as well. So this project is in collaboration with Dr. Dan Sloan at Colorado State University, and he had an undergraduate student who actually looked at these stoichiometric differences in chloroplast and mitochondrial genome copy number per cell. and found that in Arabidopsis, as you serially increase the ploidy level, there's a very clean linear relationship with the number of genomes in the organelles as well. And this occurs for both plastids and for mitochondria. And so at least one of these predictions is true. And we also have current work looking at the expression level in our lab in collaboration with Dan Sloan at Colorado State. Allopolyploidization, though, creates a special set of interesting scenarios that the cell has to overcome. So as two diploids diverge, there can be differences in the proteins that are used in the mitochondria and chloroplast, but upon polyploid formation, even though you have these two duplicated genomes from both of the diploid parents, you receive the mitochondria and chloroplast from only one of those diploid progenitors, and normally it's the maternal progenitor. And so this can create a potential mismatch between the paternal nuclear genome copies and the maternally inherited chloroplast and mitochondria, which could place some interesting evolutionary selective pressures for these genes. One way that this might work is through a process called gene conversion, where one homolog partially overwrites the other, or you can just have loss of the mismatched pair. This is actually the work that I'll be doing in my postdoc in the next year or two. But what I want to talk to you about today is just about these rates of protein evolution, and if there's a difference between the maternal and paternal copies. And so this project was co-led with Dr. Joel Charbonneau, who's now an assistant professor at the New Mexico Institute for Mining and Technology. And I have to give a lot of credit to him because this project was a beast of a project and it took a lot of work to get done. And I'm so happy that we found out on Wednesday that it's going to be accepted. So we sampled 6 different allopolyploids with varying divergence between the diploid progenitors, as you can see on the y-axis, and variable time since allopolyploidization. So as the diploids diverge, we expect there to be more and more differences, which could create more and more conflict. And as these polyploids are older, we should expect there to be more time for some of these compensatory mechanisms to actually operate. And so this involves 6 allopolyploids and their diploid progenitors and an outgroup, which meant 23 species in total, because for 2 of these systems, we used the same outgroup. And in total, this paper analyzed over 340,000 different genes. And we found these genes based on synteny and phylogeny. And we used this database called CIMER that was developed by Dr. Charbonneau and by Dr. Sloan at Colorado State University to identify which genes are used in the mitochondria, which are used in the chloroplast, and which ones may be used in both. So to set up the basics of how this test worked is we had this phylogeny of our A and B diploid genes and their respective homologs in the polyploid, which are denoted by A, T, and BT. And we also had this outgroup. And we used PAML to estimate the D and DS values on each branch of this tree. And that's kind of an estimate of how fast these proteins are evolving relative to their synonymous mutations rates. And so the dots on this plot are the values for each concatenated gene set. So we just included all the genes together and calculated the difference between, or the ratio between the paternal and the maternal DNDS values. DNDS is also referred to as omega. And we found that in 5 of these 6 polyploid species that there is a statistically significant bias towards one subgenome or the other. But we're not sure that this bias is actually real for a number of different reasons. One is that, as you can see on the phylogeny at left, the difference between the diploids and their subgenome may be different, and these diploids are not perfect models of the actual species that form the polyploid. And so it could be that phylogenetic difference could be causing some of the noise and some of the bias that we see in these data. There could also be some difference in genome annotation that could cause these problems. And then there's also this concept of homologous epistatic dominance that I had described, well, a few minutes ago before the break. that could also be describing some of these differences as well, if that's acting differently between the two subgenomes. So because there's this bias between subgenomes, we wanted to look at how these genes that are used in the mitochondria and chloroplast evolve relative to the background rate of all the other genes in the genome. And so we divided each of these gene classes into the ones that are targeted to the mitochondria and to the plastid. And we also identified genes that are targeted to the plastid but are not interacting with any proteins that are encoded in the organelles. Those genes that directly interact with genes that are encoded in the organelles, and those that directly form enzymatic complexes within the organelles. The prediction here is that the more tightly associated these proteins are with the proteins that are encoded in the organelles, the more tightly this evolutionary constraint could be affecting. And so we would expect that as we move down these 3 levels, we should see more and more significant deviations from the background rate. So all of the distributions that are colored here indicate a significant difference from the background rate, which you'll realize is only one of these distributions. Overall, we didn't see any significant difference. And even when we dug a little bit further into this one distribution, we found that it's actually just because we had a bad alignment. And so once we trimmed these sequences in the middle part of this graph here, the values really did not become indistinguishable from the background rate. And so this means that we didn't find any signatures of a global selection pressures that are acting to overcome the cytonuclear incompatibilities. We did look a little bit further down into specific protein complexes and found a couple of interesting cases that we might want to explore further, but these are actually quite rare. There's only a handful in each of these systems. And even in Brachypodium and tobacco, we didn't find anything that was significantly different from the background rate. And so these may be targets for future work to try and understand what may be driving these differences and understand why we don't see an overall global rate difference. So with that, finally, after much patience, I want to acknowledge the people in the Wendel lab, both past and present, as well as all of the co-authors that I've listed here from various universities across the university and across the world. I also want to thank my family for being here today, and for my brothers who are on the call today, and including my grandma, who passed away in 2017, but who this dissertation is dedicated to. And finally, I've made a lot of friends in grad school, and it would be impossible to thank everybody for their friendship through this time, but there are a select group of people who have helped me through specifically or through particularly difficult times in grad school. And I'm immensely grateful for their friendship when I needed it most. So thank you all for coming. Thank you all for your patience in staying with us through these technical challenges. I hope you took something away from this hour and 10-minute talk. And with that, I'm open to any questions that you may have. Thank you all.
Well, thank you, Justin, very much for that very fine presentation. I'm sure that you would be willing to entertain questions, and
Yes.
I guess you would invite people either to put the questions in the chat, or if they like, they can reveal themselves to the world and ask the question on screen.
Yeah, either one is fine with me. I don't know that I can see the chat and have my PowerPoint open, so if questions come through, somebody can read them.
As they occur.
That would be great.
Let's make sure you are.
Sure. Sounds great.
I don't want to answer the questions.
Okay.
So everybody feel free to put them in the chat or to reveal yourself and ask questions.
And I can't see you, so if you turn on your camera, I will be able to see you. I just can't see the chat.
Plenty of opportunity to rake you over the coals.
I guess I'm prepared for that.
Justin, this is Dean. Can you hear me? And may I ask a question?
Oh, I see Dean. I can't hear him.
Can anyone hear me? Hello?
We can hear you.
Everybody else can hear me. Perfect. And I can ask a question and he can't hear it. No.
Nope.
Oh, boy.
Jonathan, can you turn on your speaker, maybe?
I've got mine on.
I will keep talking until you can
there we
maybe
go.
hear me.
We've got you.
Wonderful. Awesome. Fantastic. Humbling. Wonderful are my comments first.
Oh, thank you.
So my question is this, and it deals with your, I'm not even going to try to do the M alliteration of Malvaceae, blah, 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 blah. However, your work revealed something rather interesting that I wish to ask you your perspective on, because when you got to the end of this conundrum, you essentially left us with more/better data or better methods. And this harkens back to my mind of phylogenetics for the last 65 years. And I just wanted to know your perspective, and I agree that's exactly where you are in this with respect to ploidy and trying to identify it. Where do you think the inroads will become? Will there be more on the data side or more on the analytics?
Okay. That's a great question. I think in terms of the data side, I think if we can get a better genome for Dombeya in particular, that may help clarify this. And so that may be a first start. But even if we have a Dombeya genome, though, I don't know that the methods that we currently have could even come close to understanding what the history of this polyploidy event is. Malvaceae is actually quite unique in its complexity of whole genome multiplications, because most of the events that we find on the tree are just simple duplications, maybe a triplication every now and then. But given that it's a 5 or 6-fold multiplication, there's a ton of different ways to get to that point, and I just don't think that we have the current phylogenetic methods to get there right now. So I think methods is more important, but it also can't be done without the data. I'll be a bit political in my answer. Thank you for that question, Dean.
I have a question for you, Justin.
Okay. Oh, hey, Joel.
Hi, great job. That was wonderful. You just knocked it out of the park. But I am wondering, so much about deleterious mutations is really contextual. I just thinking more about the philosophy of mutations and classifying mutations as deleterious, according to some sort of evolutionary context, in some way divorces it from the ecological context in which the mutation occurs. And I wonder if you've thought about this at all, and if there's any hope that we can merge those two at some point. Thanks.
Sure. So I think you're asking kind of what role that if local adaptation could also play a role in the inferences that I made. And I think certainly these comparative methods only look at historical conservation. And so it could be the case that a new mutation that historically has been deleterious may actually be really adaptive in a new environment. And so I think it's certainly the case that locally adapted alleles could be present in this deleterious mutations case, and actually could be due to some of the load that we find in the Hawaiian cottons. And I think also just using raw or just using population genetic methods instead of comparative genomic methods could be a really nice complementation to this as well. Unfortunately for the case in cotton, there's so much species divergence that you can't really use population genetics to look at the difference between the polyploids and the diploids. But in some of the more recently formed polyploids, you can certainly look at some of the ecological differences and do and look at how different mutations may have different selective coefficients. There was a paper recently published by Ryan Gutenkoontz at the University of Arizona that actually developed a method to look at the joint distribution of fitness effects. So in two different populations, you can kind of look at the correlation between mutations in one population versus the other. And I think an approach like that could be really helpful in exploring this question even further from a population genetic standpoint, and to kind of circumvent the challenges that this comparative genomics technique brings.
Wow, that was really insightful. Thank you so much.
Thanks, Joel. And thanks for all your help on this project too. I really appreciate it. You're great.
I didn't do anything compared to you.
Are there any questions in the chat?
Roger Wise wished to add a historical factoid that the lead postdoc on early slide you showed on main sequencing was Fouché Wei, another ISU IG student.
Oh.
IG was IgG.
Fantastic. Yeah. So yeah, the first maize genome was published by Pat Schnabel, who's a professor here at Iowa State in the gardening department. Yeah. Yep. Yeah. At the University of Nebraska. Yep.
So are there any more questions from the global community that's tuned in today?
I heard my brother Chase was going to have a question, but I'm not sure if he's gun shy or not.
Sure, I can fire away. Hey, great job. I'm proud of you.
Thank you.
Back, you were talking about the theory of the 1932 book, and you made a comment there and was curious how much of your research went into this. But you said the theory was almost older than sliced bread.
Yeah.
About how old do you think sliced bread is?
Sliced bread was invented in 1928 in Chillicothe, Missouri.
Yeah. Pretty close then.
That's not pretty close. That's the correct answer.
Well, they're about the same age. You're right. It is about older.
Coming from a brother, I'm just glad that question was appropriate. All right, well, I think maybe we will end this part of the defense, which is the public part. And after a short break, we will reconvene with just the committee,
Sounds great.
including the one committee member who's zoomed in from Colorado, Professor Nason. I'm sorry you didn't get.
Sounds good. Well, thank you all for joining today. I really appreciate it. Thanks for all your help.